Hey everybody, hope you all had a wonderful Thanksgiving break. Thank you for joining us for the penultimate TiMM presentation. This week, Carson, Maggie, Margot, Sam, and I are excited to share with you a pair of papers that look at the relationship between interferon signaling and COVID. Before we delve into the details, I just want to go over what we'll be talking about for the next half hour. I'll begin by giving some background information on interferon signaling, specifically its components and effects. Maggie will then enlighten us with a few clinically relevant cases, before Carson and Sam tackle the methods and the results. Finally, Margot will finish it off with a discussion of the implications of these two papers. To start, most of you should be pretty familiar with the image on the left, which was pulled from one of our immunology slides. Canonically, the innate immune system has evolved a constellation of pattern recognition receptors to detect dangerous materials in the molecular milieu of the host. These are the PAMPs and DAMPs that we've talked about in class. Especially in the context of viral infections, a common pro-inflammatory cytokine that's often elicited is interferon. You can also note from the figure that multiple cell types of both arms of the immune system contribute to interferon signaling. While we'll be primarily focusing on type 1 interferon, interferon actually comes in 3 different flavors. Type 1 interferons are often produced by monocytes and fibroblasts and play a crucial role in preventing viral dissemination. And we'll dissect this a little bit more in the next couple of slides. Type 2 interferon consists of interferon gamma and is usually released by type 1 CD4s and ultimately polarizes immune response towards a Th1 response, which is more centered around cytotoxic killing of cells infected with intracellular pathogens. Finally, interferon lambda was recently explored and implicated in both viral and fungal protection. There are a couple of essential components in interferon signaling induction. As mentioned previously, pattern recognition receptors such as TLR3 detect double-stranded RNA and can activate IRF3, a transcription factor that induces the expression of interferon alpha and beta. Released type 1 interferon is detected by IFNAR1 and 2, the interferon receptor, which subsequently induces STAT signaling. Ultimately, together with IRF9, STATs form a complex that activates interferon-stimulated genes, also known as ISGs. Interestingly, the ISGs also include IRF7, which promotes further type 1 interferon production, creating a positive feedback loop. So what do these ISGs actually do? While many viral defenses are attributed to them. For example, ISGs induce upregulation of MHC molecules to enhance immune detection and antigen presentation. The former is essential in the antiviral arsenal, as cytotoxic killing of virally infected cells is often mediated by TCR engagement with MHC class I proteins presenting viral intracellular peptides. ISGs also increase levels of RNase L to directly destroy viral and intracellular RNA. This is further coupled with protein kinase R, which phosphorylates and cripples initiation factor 2. This essentially ensures decreased protein synthesis in an infected cell, slowing potential viral replication. Finally, ISGs also increase activity of p53, promoting apoptosis and limiting viral spread. On a side note, there is also increasing evidence that interferon is involved in suppressing angiogenesis and reducing tumor size. Given these interferon antiviral functions, it is unsurprising that individuals with inborn mutations of components in the interferon signaling pathway exhibit compromised protection against viral infections. Maggie will show us examples of this in the following section. Interestingly, evolution has also primed many viruses to avoid the immune system by sequestering or destroying components in the interferon pathway, making pathogens like HPV and CMV particularly difficult to clear. Finally, studies have shown that type 1 interferon induction is important for vaccine efficacy, and perhaps this is something to think about with the upcoming COVID vaccine. In addition to inborn mutations in the proteins of the pathway, interferon signaling can also be compromised via autoantibodies. From class, we talked about molecular mimicry as a virulence factor for many bacterial pathogens. For example, M protein of strep pyogenes is similar to endogenous proteins in the body such as myosin, and is an underlying basis for rheumatic fever in patients with untreated strep. One can easily imagine that autoimmune diseases such as APS1 can lead to production of autoantibodies against type 1 interferon, effectively sequestering and neutralizing the molecule, as shown on the figure on the left. Cases like these often phenocopies those with inborn mutations in the interferon pathway, and, as demonstrated in the papers for today, are often associated with life-threatening COVID. So why does any of this matter? Think about that as Maggie presents some interesting and relevant clinical cases.
Now, I'll introduce a few clinical cases that illustrate what happens when one of the pathways Ming discussed isn't functioning properly. For time's sake, I won't always delve into specific mutations or inheritance patterns, but I will share links to all of these papers in the discussion. First, we have a case of a 2-month-old boy of consanguineous ancestry with a digenic inherited immunodeficiency. He had homozygous deletions in IFNGR2, which encodes the signaling chain of the interferon gamma receptor, and IFNAR1, which encodes the signaling chain of the interferon alpha receptor. He presented with a fever, disseminated mycobacterial infection, strep viridans bacteremia, and severe cytomegalovirus viremia. Due to the importance of interferon gamma in macrophage activation and response to intracellular pathogens, the IFNGR mutation made the patient susceptible to disseminated mycobacterial infection. In fact, this infection is characteristic for patients with inborn or autoimmune-mediated IFNGR deficiency. The IFNAR mutation led to minimal expression of type 1 interferon-stimulated genes. Unable to mount a sufficient response to CMV, the patient developed severe viremia. In 2014, Lim et al. Published a study on 6 pediatric patients with herpes simplex encephalitis, or HSE, who had missense TLR3 mutations. As we recently learned in Micro, HSE is a life-threatening complication of herpes simplex virus 1 and is characterized by acute necrotizing inflammation of the brain. Infection often begins with fever, headache, seizures, and confusion. Because damage is typically localized to the temporal lobe, as seen in the picture, patients may suffer from symptoms like aphasia, which is a diminished ability to communicate using words. HSE can recur after convalescence, and TLR3 deficient patients are more prone to relapse. All TLR3 mutations in this cohort led to loss of function. As a result, patients' fibroblasts displayed increased susceptibility to herpes simplex virus. Previous studies also suggest that TLR3 deficiency increases viral susceptibility in neurons and other brain resident cells. In 2019, the same research team published a report on 3 unrelated pediatric patients who developed acute respiratory distress syndrome, ARDS, upon infection with influenza A virus. ARDS occurs when fluid builds up in the alveoli, impeding gas exchange. This causes severe shortness of breath, confusion, and fatigue. It is life-threatening. The researchers found that TLR3 mutant leukocytes produce normal levels of interferons in response to flu. This makes sense because TLR3 is often absent or redundant in these cells. However, mutant fibroblasts and iPSC-derived pulmonary epithelial cells produced insufficient levels of interferon-stimulated genes. The TLR3 roadblock in interferon signaling led to the patient's severe influenza disease course. IRF7 deficiency causes an influenza disease course similar to what we just saw. In this case, a 2.5-year-old girl presented with acute respiratory distress syndrome upon infection with H1N1. She was found to have a compound heterozygous mutation for the IRF7 gene. The patient's leukocytes and plasmacytoid dendritic cells, or PDCs, produced very low levels of type 1 and type 3 interferons in response to H1N1 and herpes simplex virus, leaving her vulnerable to severe infections. The last disease we'll discuss is autoimmune polyendocrinopathy syndrome type 1, or APS1. This disease illustrates the effects of anti-interferon autoimmunity, as well as autoimmunity in general. The symptoms of APS1 emerge early in childhood, and the most common is chronic mucocutaneous candidiasis, or yeast infection. Interestingly, chronic candidiasis is also seen in patients with myasthenia gravis and thymoma, who also present with anti-interferon antibodies. When APS was first discovered, this clinical detail provided a clue as to its possible etiology. The underlying cause of APS1 is a mutation in the AIRE gene. This codes for a protein that helps destroy autoreactive T cells before they exit the thymus. All APS1 patients express autoantibodies against type 1 interferons. Neutralizing antibodies mimic the effects of a pathway deficiency, and patients are subsequently prone to chronic yeast infections and other sequelae. Autoreactive T cells can also target organs or tissues, leading to symptoms such as alopecia, hepatitis, and vitiligo. And now, we'll shift our focus to COVID-19. The big question we want to ask is, why do some seemingly healthy people get very sick or even die? We will now explore how latent immunodeficiencies and autoantibodies may contribute to severe COVID-19 disease course.
Now that we understand some of the background information on the interferon pathway and interferon deficiencies, we will jump into the first paper discussing the role of host genetics in susceptibility to severe COVID infection. The goal of this paper was to look for the accumulation of damaging mutations in genes known to be important for viral defenses. This project was led by two of the world's leaders in the role of host genetics in susceptibility to viral infections. Jean-Laurent Casanova is an icon in the field of primary immunodeficiencies. He is a Howard Hughes Medical Institute investigator who has a lab at the Rockefeller University, as well as a dry lab at the Imagine Institute in Paris. He teamed up with his friend and colleague, Helen Su, who runs a lab in NIAID in Bethesda. Helen is one of my mentors and let me help out with this project as I wrapped up my gap years at the NIH. Thus, I have an inside perspective into these two papers. Back in March, when very little was known about COVID, Helen and JLC put their heads together and realized they could provide important insight into the mechanism of immune defense against COVID. They created the COVID Human Genetic Effort, an enormous international consortium whose goal was to sequence as many COVID patients as possible, as fast as possible. To illustrate this point, here is a map showing just the sequencing hubs within the consortium. This was a remarkable effort that was put together in just a few weeks. The central idea of the human genetic effort is the idea that very rare genetic variants tend to have a large effect. The typical population genetics approach, using GWAS, is only able to pick up common variants that have only a small effect. We thought one single variant could be sufficient to cause disease. To find these rare and often private variants, we needed to conduct whole exome or whole genome sequencing. Then we looked for predicted loss of function variants in genes known to be important in the type 1 interferon response to viral infection. To review what we just learned in epidemiology, this was a case control study in which we counted up the number of patients with a predicted loss of function variant in the pathway given their disease severity. To analyze the results, we make a 2x2 table. When we make this, we see that the odds of having a rare variant given severe disease is 9 to 650, while the odds are only 1 to 533 given mild disease. This gives a statistically significant odds ratio of 8, suggesting an association between these variants and severe disease. Next, we asked if there was an accumulation of loss of function missense variants in the severe COVID cohort. We tested the function of all 109 rare, non-synonymous variants in the pathway, as well as the 9 predicted loss of function variants, using an in vitro reporter assay. As expected, all 9 predicted loss of function variants were indeed loss of function. Additionally, we found 15 rare missense variants with reduced activity. In total, we found 24 loss of function variants in the severe disease cohort, including 4 autosomal recessive and 19 autosomal dominant cases. 3.5% of patients with severe COVID infection in our cohort have a genetic deficiency of type 1 interferons. As we conclude our discussion of this manuscript, I will leave you with a few questions to consider as you prepare for our discussion tomorrow. First, the purpose of the study was to look for enrichment of rare loss of function variants in severe COVID patients, that is, enrichment when compared to mild COVID patients. With this in mind, what are the reasons for and implications of not including in vitro testing of rare missense variants in the mild COVID cohort. Second, the paper's conclusion is that 19 of the severe cases have autosomal dominant loss of function disease. The two models to explain this phenomenon would be dominant interference or haploinsufficiency. Was enough data shown to convince you that these patients do indeed have an autosomal dominant disease? Last, we know that loss of function mutations in the type 1 interferon pathway are bad. Maggie discussed several diseases in this pathway that lead to susceptibility to severe infection from common pathogens, such as the flu and CMV. Why then would a person with a type 1 interferon defect be healthy for decades until having severe COVID? Why would he not have a significant history of hospitalizations for viral infections? The party line is that diseases such as the flu are so common that we always have a partial immunity to new infections because of our past history. Thus, our immunological memory may be able to compensate for an impaired interferon response. However, since most humans have never been infected with a similar virus to SARS-CoV-2, we have no immunological memory. Thus, we need our immune system firing on all cylinders, so to speak, and will get a severe infection if there are any defects in our response. I'm very interested to hear if you think this is a compelling argument.
We'll now shift gears to talk about the second paper, which is about autoantibodies against type 1 interferons in patients with life-threatening COVID-19. So we just talked about inborn genetic errors, somatic mutations, and we generally think of inborn somatic mutations as leading to loss of function through leading to errors in mRNA, which results in non-functional or even truncated proteins, and that's how you get a loss of function. But as it turns out, it's also possible to get loss of function when all of this stuff works perfectly and you get perfectly folded functional proteins. Then a neutralizing antibody comes along and neutralizes the function of the protein. And so in this case, the well-folded protein would be a type 1 interferon, and this antibody would be an autoantibody or a self-antibody that is present in the immune system that neutralizes the function of the type 1 interferon. These autoantibodies, as Meng talked about earlier, can compromise host immune function and can increase the susceptibility to opportunistic pathogens. And so to demonstrate this, one case of a particularly opportunistic pathogen, a non-tuberculosis mycobacterium infection, you can see that when you take serum from a patient who has this rare infection, and you put it on cells in vitro that you stimulate with interferon gamma, the control cells get super stimulated and secrete a ton of TNF-alpha. But when you put on patient serum that we know contains autoantibodies, the function of the interferon is compromised. And similarly, we know of this disorder called APS1 that is caused by a genetic mutation in this gene called AIR, or autoimmune regulator. And these guys all get the opportunistic infection candidiasis, and it results in autoantibodies against the type 1 interferon. So not interferon gamma, but rather interferons alpha, beta, and omega. And so our story begins when the authors admitted 3 patients with APS1 to the ER who had life-threatening coronavirus disease. And so it led the authors to ask the question, well, are the autoantibodies causative of severe disease, and is it possible that other people without APS1 could have autoantibodies, and could that be a driver of their severe disease? So they harvested plasma from healthy controls, folks who were infected but had mild or asymptomatic COVID-19, and severe COVID-19 cases. And what they did was they incubated these folks' plasma with this pool of beads. There's 18 different kinds of beads. Each bead is a different color fluorescently, and you can discriminate this on flow cytometry. And each color bead is linked to a particular cytokine, like interferon alpha or beta or omega, some of the interleukins, etc. You incubate patient plasma with these beads, and in this way, if the plasma contains an autoantibody, it gets stuck to the bead that's specific to the protein that the autoantibody targets. You then can detect the presence of antibodies with a secondary antibody. You run these guys on flow. You're gating each bead type, and then you're pulling out all of the beads of one type and looking at the PE intensity to see how much of an autoantibody exists. And so the authors did this, and they found a striking prevalence of about 15% of patients with life-threatening COVID had autoantibodies against either interferon alpha or omega, or both, whereas those who were asymptomatically infected and healthy controls had a much lower prevalence of these autoantibodies. Importantly, want to understand if these autoantibodies have some sort of function that antagonizes type 1 interferons. And so a couple of good demonstrations of this were in vitro. In this case, the authors are stimulating cells in vitro with either interferon alpha or interferon omega in the presence or absence of serum. And so here in the presence of control, healthy serum, you can see a stimulation of cells and an induction of phosphorylation of STAT1 with either of the two cytokines. But when you put patient serum onto these cells, it abrogates the ability of these interferons to induce the phosphorylation of STAT1 in these cells. And importantly, this is dependent on an immunoglobulin G, because when you deplete IgG from the serum, you lose the ability to do this. So what this tells us is that there is an IgG antibody in this patient's serum, and that has the ability to antagonize the interferons, and particularly the type 1 interferons. And this also translates to the context of viral infection. You can see here is an assay for infection of these HUH 7.5 cells in vitro in the presence or absence of serum, and using an interferon alpha-2 monoclonal antibody, and then in the presence of interferon alpha-2. So when you give these cells interferon alpha-2, the blue curve shows that they're markedly resistant to infection with SARS-CoV-2, but as soon as you drop patient plasma on them, the patient plasma sops up all this interferon alpha-2, presumably due to the autoantibodies, and now you can get really robust infection. And lastly, another interesting observation from the paper was the demographic distribution of these autoantibodies. The autoantibodies were really, really enriched among men in the cohort. So almost everybody who had autoantibodies was a man. And there was a less pronounced correlation between the age of onset of the autoantibodies. So it seems like the older you are, the higher risk you are for having anti-type 1 interferon autoantibodies. So some discussion questions, just some things to kind of mull over. This bead-based flow assay is really interesting. The paper is pretty straightforward, but maybe start thinking about other types of clinical questions that you could answer using a protein-based, bead-based assay like this. And then the authors claim pretty strongly that these type 1 interferon autoantibodies are causative of severe COVID-19. Do we think this claim passes muster? Think about this a little bit. Remember the Bradford-Hill criteria that we talked about in epidemiology, and ponder about whether the authors were able to find any patients with mild COVID-19 who had autoantibodies. That would kind of undermine sufficiency.
We will now briefly discuss the clinical implications of the findings from these two papers. In a recent interview, one of the investigators in these studies stated, "It would be important to implement routine testing for autoantibodies in all patients infected with COVID-19 to be able to implement adequate treatments and preventative measures, and perform genetic testing in the patients with unexplained severe COVID-19. If screening occurs early in disease progression, we can hypothesize that patients will benefit from guided treatments, such as the administration of interferons. For example, results from these studies suggest that early treatment with interferon alpha is unlikely to be beneficial. However, autoantibodies against interferon beta appear to be rare, suggesting a potential beneficial effect in these patients. However, with screening comes several logistical and ethical considerations. How do we decide who gets screened for autoantibodies? Will it be everyone that tests positive for COVID-19, or only in severe inpatient cases? Can we use this method to treat severe cases or to prevent them altogether? What about the feasibility of performing genetic screens? Should this be reserved for the most severe cases? And if so, has the disease progressed too far at this point? You can imagine the complexity here. Additionally, one other tangible application is the testing of convalescent plasma, or the method of serum transfer from recovered COVID patients used to prevent or treat others. Perhaps selecting for donors without autoantibodies would result in a more efficacious treatment. So we know that interferons are crucial for antiviral immunity, but how have they fit into COVID-19 as an actual treatment strategy? This already outdated schematic demonstrates the rapidly evolving landscape of clinical trials ranging from antimalarials to steroids to HIV medications being investigated for their efficacy in treating COVID-19. As you can see, interferons make up only a fraction of the global effort. We did want to mention a series of trials where interferon treatment is currently being investigated. These trials are called the Adaptive COVID-19 Treatment Trials, or ACT, which are multi-center, randomized blinded control trials that are testing the safety and efficacy of different therapeutics for the treatment of COVID-19 in hospitalized adults. Trial 1 featured remdesivir, which binds to the viral RNA-dependent RNA polymerase, inhibiting viral replication. In early November, the New England Journal of Medicine published the final report from Trial 1, which concluded that remdesivir was superior to placebo in shortening time to recovery in adults who are hospitalized with COVID-19. Subsequent trials are aimed at investigating the therapeutic effects of combination therapy with remdesivir plus another intervention, baricitinib, a JAK inhibitor in Trial 2, and interferon beta in Trial 3. As of November 19th, the FDA has already approved the combination therapy and remdesivir alone for the treatment of COVID-19, as results from these clinical trials has shown reduced time to recovery. A few days ago, Trial 4 was announced, already making these slides out of date, which will compare remdesivir plus baricitinib or dexamethasone, a corticosteroid used to treat inflammation, such as that seen in arthritis. Over the past 9 months or so, we have seen an astounding number of clinical trials being conducted across the world, some showing promise while others falling flat. We have seen the influence that news and media outlets has on the public knowledge of coronavirus treatments, and how some treatments are not backed by rigorous testing in the lab and through clinical trials. We have seen this with the use of the antimalarial hydroxychloroquine. We have also seen promising trials fail to show significant clinical effects, such as the HIV medication lopinavir or ritonavir. Discord between the FDA and WHO is even calling into question the efficacy of remdesivir due to heterogeneity in trials across countries. Furthermore, results from the ACT trials have not shown huge mortality benefits. Rather, they shorten time to recovery by the span of a few days. The point here is that as future physician scientists, we need to think critically about how research is conducted, what amount of evidence we deem necessary for significant conclusions, and what that means for our own practice. We'll leave you with a few discussion questions for our session on Wednesday, and we encourage you to think about these broadly. We know that not all the severe cases were explained by these inborn errors or autoantibodies, so we wonder what else might be underlying severe COVID-19. Are there other interferonopathies? What might you think be going on here? Also, one cool thing about these papers shows the effort and collaboration underlying a lot of COVID-19 research. How do you think COVID has changed the way we do science? And what changes do you think will last? We briefly touched on some of these clinical implications, such as how would you choose whom to screen according to these studies? What experiments or clinical trials might you want to design moving forward? And importantly, what amount of evidence is needed for sound conclusions? And what are meaningful results, meaning reduction in recovery time versus actually reducing mortality due to COVID-19? Does knowing that there are other studies published also showing type 1 interferon deficiencies in severe patients help you believe the author's conclusions? We're looking forward to our discussion this Wednesday, and thanks for listening.